All right, we're going to look at our last section on passive transport. Let me get that ready for you. Went a little bit too far, sorry about that. Here's where we ended up on the last slide was we talked about osmosis drawings. And now we're going to enter that last section on passive. Remember, passive requires no energy. Passive moves from high concentration to low concentration. So the first thing we're going to look at is an ion channel. And an ion channel is a donut-shaped transport protein with polar pore. So remember, polar means that it has positive and negative poles to it, through which ions can pass. These pores can open or close by stretching the membrane, electrical charge, or binding certain molecules to it. And on the bottom here, you can see on the picture, there is an open ion channel, and then there's a closed one. So it will change its shape once a molecule binds to it. Now, depending on the concentration between my high and low will depend on how fast things will diffuse across the cell membrane. If it is extreme, so like on that bottom one, it says if a cell is very negative and the outside is very positive, it's going to move really, really fast. And if there isn't much difference between positive and negative, it's not going to move as fast. There are certain ions that are used in these channels. And so the top three I give you, sodium ion, potassium ion, and calcium ion, are considered cations. Now, if you remember back to chapter 2, an ion is a particle with a charge, and a cation is a particle with a positive charge. So you can see sodium, potassium, and calcium all have positive charges. Sodium, potassium has a +1 charge. Calcium has a +2 charge. The last one I'm giving you is chlorine, and chlorine has a -1 charge. It is still an ion, but it's no longer a cation, it's an anion, a negative ion, A-N, a negative ion, anion. And so this is one of the examples of a negative ion. And all four of these are used in ion channels. Now, these ion channels can cause some problems. So if there's a malfunction of your ion channel, one of the possible diseases that you could have is cystic fibrosis. And cystic fibrosis will really affect especially the lungs of an individual. And we'll talk about cystic fibrosis later on when we talk about genetics. And then another thing is my good buddy Novocaine when I have to go to the dentist and get a cavity removed. And they bring out this huge needle. And I always cringe when I see this needle. In fact, I have a picture of the needle down below. And they give it to you, and it is a little painful, and you feel a little pressure. And then all of a sudden, you don't feel anything on that side at all. And you can still carry a conversation with your dentist, even though you're really numb. The point of how Novocaine can work is it actually blocks the ability of some of these ion channels to work. And so that message from those ion channels are not reaching it into the nerve cells, and they're not able to spread that message then up to the brain that, ouch, this is really painful. You should be screaming right now. And we're really lucky that we live in this time and day where the dentist is not as painful of what it used to be. The last section that I have, so this is the last thing we have, is on carrier proteins. If you remember, if you have a small particle, they can easily go across the cell membrane. So really, really tiny molecules have no problem. Big molecules have an issue, and this is where you might need a carrier protein. And amino acids are big molecules. Sugars are big molecules. So they need a little help. And so we have these buddies called carrier proteins. And you can see the picture, and we'll kind of describe the steps on the next slide. But here's my carrier protein. The white part is the inside of the cell. The blue part is the outside of the cell. More carrier proteins on the outside than inside. So higher concentration outside, lower concentration inside. And remember, passive transport move from high concentration to low concentration. So I'll have one of these molecules hit the carrier protein. And what happens is it causes a change in the shape of the carrier protein. And that shape will then allow, it actually an opening to the inside of the cell, whereas beforehand it had an opening to the outside of the cell. And so these carrier proteins will help move amino acids or sugars into the cell. All right, so we're going to go through the steps. I kind of went through them already, but now we can get them formal in notes. A carrier protein changes its shape for a molecule or changes its shape for a molecule. So depending on what molecule, and these are going to be molecule specific, the new shape exposes the molecule to the other side of the membrane. And I kind of showed that where it was closed, then all the inside, and then once that molecule attached, it then opened to the inside. And proteins protect the molecule from the interior of the cell membrane. And so that's why it's a little bit protected as it enters into the cell. It will be in the form of a vesicle eventually and move its way, probably like if we're talking about it being a sugar molecule, probably to the mitochondria. So that way it can be broken down and used for ATP. I'm excited to see this. This means that we've made it through the first half of the section. So the next video I will have will actually start the rounds of active transport. This is the last video on passive.